Hello, and a very warm welcome to everyone joining us today. My name's Charlie Carter, and I'm delighted to be moderating today's session titled Automate to Accelerate: How to Streamline Single-Cell Sequencing with Automation. I'm joined by our guest speakers, Dr. Vasudha Nair, Scientist of Cellular Sciences at Opentrons, and Cooper Seatman, Technical Sales Manager at Parse Biosciences, who will be discussing how to efficiently automate your whole sequencing workflow from fixation to library prep. Please feel free to submit any questions you may have during the webinar today. To do this, simply head to the engagement window to the right of the auditorium and click to open the Q&A window. So, without further delay, I'm delighted to hand over to Vasudha and Cooper, and I would like to thank them again for presenting to us today.
Well, thank you so much for joining us today. I'll start with just a brief introduction of who I am, and then we'll dive into it. So my name is Cooper Seatman. I'm the Senior Technical Sales Manager at Parse Biosciences for the Central US as well as Canada. And I'm here today to kind of give you a brief introduction to our Evercode platform presented by Parse. So starting from the top and where Parse came from. So our two co-founders came out of the University of Washington in 2018. They started the company that same year. Took 3 years to really optimize the kits to make them commercially ready to roll at 2021. And we've been out on the market for 4 years now, and we've seen rapid adoption. So 2,500 plus labs are using Parse globally. We also are excited to share that we are in over 150 plus publications, and we have plenty of customer data sets, really just showing the snowball effect of validation, which is great for both us and our end users, that we are a validated technology and people are excited about the benefits it can provide. So looking at the single-cell RNA-seq space in general, what we're finding is studies are getting larger and larger. So we're seeing more RNA-seq publications and more single-cell publications in general year over year. Not only that, but we're seeing a greater share of those RNA-seq publications are coming from the single cell space. So now we're seeing more single cell publications are getting out there year over year, but not only that, we're seeing more cells per publication. So people are increasing the number of cells they're analyzing. Up till 2022, it's relatively modest increase, but since 2022, for a variety of reasons, we've seen a pretty rapid growth in number of cells, being at over 100 cells per publication in 2024. A couple of reasons for that is I think technologies like ours enables you to scale up, but also sequencing is getting more affordable. And why are people looking at more single cell publications and larger data sets? It's to give them more information. So more statistical power, more replicates, more time points, more patient samples. But looking at this graph here, it shows kind of the direct correlation of analyzing more cells and the amount of different cell types that you can detect. So getting more rare cell types, which can be important in a lot of these biological fields. So shifting gears and looking more at the Parse Evercode technology itself, we provide different kit sizes. And these different kit sizes give you a total output of cells. So the way I want you to think about it is you have a total output of cells. You can divvy up those cells to really however many samples you see fit. So looking at that 100,000 cell kit, the WT kit we have in the middle, you have 100,000 total cells. You could divvy that up to anywhere up to 48 samples. So many people will start, let's say they do 10 samples. You could run 10 samples on the 100,000 cell kit and capture 10,000 cells per sample. Same thing as you scale up with the million cell mega kit. You can now run 96 samples at 11,000 cells per sample. So it gives you the flexibility to kind of run however many samples you see fit, whether it's high throughput in the number of samples or high throughput in the number of cells per sample, depending on your research question. What we actually just launched 2 weeks ago is our new WT Penta kit. So we're really excited about this kit because it gives you more scale, right? We just talked about how single cell publications are increasing in the number of cells that are being analyzed, and we're following that trend and showing you that you can scale this up and even further and run 5 million cells in a single run. Today, most of the talk today will be talking about our whole transcriptome assay, but we also have other profiling kits that are available as well. So we've got immune profiling, we've got pooled CRISPR screens, we've got targeted gene panels, but for the majority of today, we'll talk about our bread and butter whole transcriptome assay. Okay, looking at the complete solution and really what we can provide here at Parse, we start with that single cell, high quality single cell or single nuclei suspension. From there, we're going to jump into our fixation. So this is where Parse comes into play, where you're able to fix and store these cells for up to 6 months. Once you have all your cells or nuclei ready to go, then you'll jump into the barcoding and library prep process. Once you're done with the library prep, you're going to end up with Illumina libraries that you can send off for sequencing. These libraries they're set up to be run on Illumina, but they can be run on any of these other sequencing platforms that are available on the market. After you get those sequencing data files back, you can then plug it into our TrailMaker data analysis package, which is free with the purchase of the kit. And then lastly, which is obviously very relevant to today's discussion, lending itself to this complete solution, we wanted to bring automation solutions to the table to automate a lot of these manual processing steps in the front end. So the fixation, the barcoding, and library prep. So we'll talk about that with Opentrons in a little bit here. But diving a step deeper and kind of looking at the technology itself, what happens is we start with that single cell suspension, and then we're going to fix and lock that gene expression within the cell. From there, we're actually going to permeabilize the cellular membrane. And what this allows for is these barcodes and enzymes to enter the cell, use the cell itself as the container for the reaction. So as opposed to emulsifying a cell in an oil droplet, we're actually using that cell itself, and we're keeping the cell intact throughout the processing, which means there's no cell shape or size limitations in our assay. At this point, we've got millions of fixed permeabilized cells. How do we get to that single cell resolution without a microfluidic instrument? The way we do this is combinatorial barcoding. So I'm going to walk through the process step by step, but essentially what we're doing is it's a probability approach of taking these cells through a unique pathway, ending up with their own unique identification. So I'll walk through it and it'll make more sense in a second. So what we have here is a 96 well plate. Each well in this plate contains a barcode unique to that well only. So we're going to evenly distribute our millions of fixed permeabilized cells across all 96 wells. And you can see zoomed in on this blue well here, all of the thousands of cells in that blue well receive blue barcode 1. So now all of the cells have a barcode, but many have the same barcode, right? So it's not single cell resolution quite yet. So what we actually do is we take all of the wells, pool it into one giant population, mix it up, and then repartition with a multi-channel pipette into another 96 well barcoded plate. So now there's new populations of cells in each well. And you can start to appreciate that naturally through this processing, these cells will take a unique path through each of these 4 steps, ending up with their own unique identification combination of 4 barcodes. So you do another pooling step into another 96 well plate. Now the cells have 3 barcodes, one last pooling step into 16 sub-libraries where the library preparation will occur. So in the end, there's 14 million unique pathways that the cells can travel through, which gives us the confidence to say you can capture 1 million uniquely identified cells via that combination of 4 barcodes. One last thing I want to highlight on this slide is sample multiplexing. So sample multiplexing is actually very straightforward in a Parse assay. There's no custom barcoding needed. It's all built into barcode 1. So barcode 1 is also our sample identifying barcode. To keep it simple for this mega kit example, let's say you have 96 samples. You're actually going to put 1 sample into each well in this first plate. And then bioinformatically, we go back to barcode 1. We know exactly which cell came from which sample because we know which samples in each one of these wells on the first plate. So it's very straightforward, and we have a loading table that tells you exactly how to load that first plate, depending on how many samples you have and how many cells you want to analyze. So you end up with this process, you end up with those 16 sub-libraries that you can send off for sequencing. From there, you're going to get those fast queues back, and that is where you can plug it into our cloud-based TrailMaker pipeline. So the TrailMaker pipeline has 2 different modules. It's got the pipeline module and the insights module. So starting with the pipeline module, you'll see here you have your standard RNA-seq analysis. QC, you're going to see how many cells, how many genes, how many transcripts you're getting. You're also going to be able to get the standard output reports. So you're going to get your count matrices that you can plug into Syrah or ScanPy for your custom analyses. And then you move to the insights module. So the insights module will actually allow you to to customize any filters. You can add that biology or the metadata to your data, your sequencing data. So you can look at the differential gene expression among different populations you're interested in. You can zoom in on different clusters, annotate, and even get to publishable plots and figures. So kind of wrapping up the Evercode technology in general and looking at some of the benefits and what makes us different out in the space is, one is that scalability. So we're able to scale up to 5 million cells as of last week and 384 samples in a single run. So it gives you the ability to really grow your experiments and run more replicates, more time points, more conditions, more samples in general. Number two is there's no instrument required. So again, we just walked through the assay. It's relatively straightforward. And essentially, if you purchased a Parse kit tomorrow, you could run it next week. It also lends itself really nicely to automation because there is no custom microfluidics or any custom instrumentation needed to build into that automated system. Third thing here is the unmatched data quality. So we're finding we're detecting more genes and transcripts per cell and seeing less doublets and less ambient RNA noise compared to droplet-based methods that are currently out there. And then fourth is the power of the fixation. So this fixation will allow you to fix and store your samples, take complete control of the experiment before running all of those fixed samples through the barcoding and library prep process. What that can do is dramatically decrease the batch effect because now you can compare sample to sample without having to run those live cells when they come as batches. Shifting gears here and talking a little bit about why automation and why it fits so nicely with Parse. One is that predictability, right? So what we wanted to do is test a manual workflow with Parse along with an automated workflow. And what we found was very similar cell populations, minimal variability, as well as high correlation when looking at the non-automated manual workflow with the automated workflows. And we'll talk a little bit more about that when Opentrons kind of chats through how we've adapted Parse to be worked on the Flex instrument. The other aspect of this is the scalability. What it can do is it can drastically decrease the hands-on time. As you saw when we were talking through barcoding, there are quite a few pipetting steps. And what this automation will allow you to do is be hands-off for those pipetting steps. So the lab techs can be doing other things in the lab and just creates a more efficient process. So in general, I think overall, what this automation with the Parse workflow provides is taking you from that very active pipetting workflow to very passive efficiency, right? It's more predictable. It minimizes the variability. It also gives you the ease to scale this thing up pretty dramatically with lower failure rates, as well as the cost effectiveness, right? Less hands-on time, less manual labor, also less repeats, and in general, less human error caused when working with automation. So with that, I'm going to pass it over to Visuddha over at Opentrons to talk through how Parse is implemented on the Opentrons system and how we can kind of benefit your scalable workflow. Thank you very much.
Thank you, Cooper, for the excellent presentation on the Parse Biosciences Evercode technology for single-cell sequencing. Now I would like to shift the focus to how we at Opentrons approach automation integrating the Parse technology. So I want to start off with a quick look at the mission of Opentrons, which will provide context for why and how we approach automation. Historically, automation in laboratories has been limited to well-funded and high complexity research facilities, creating barriers to broader adoption. At Opentrons, our mission is to democratize access to laboratory robotics, making automation more scalable, cost-effective, and widely available. To achieve this, we have developed the Opentrons Flex platform designed for complex workflows, including the single-cell RNA-seq. This is a slide showing the core capabilities of the Opentrons Flex platform. So, the Flex platform itself is modular, benchtop, and comes with either the 8 channel, the single channel or the 96 channel options, and features modularity that allows users to scale their setups. Users can select from different parse workflows and start with the mini, scaling up to the penta on the same platform. So stepping back, it's important to consider why automation is valuable for single-cell RNA sequencing. As Cooper highlighted very well, that researchers are increasingly needing to process large number of samples and cells replicates in their studies. So with this growing scale, manual sample preparation becomes more time-consuming, labor-intensive, and prone to variability. On the Opentrons platform, users can automate key steps of the Evercode workflow right from fixation to the library through to the library prep. And that way we can avoid any variability that happens during a manual workflow. So thus, what happens is there is an enhanced efficiency and scalability, improving consistency and reproducibility. So as you see in this slide, automation is not plug and play, requiring expertise, but the platform users, so the platform includes user-friendly features to ease the learning curve. We have simplified the process to take away much of the complexity and effort involved in automation. So when you think about this workflow itself, it really comes down to two main steps: selecting and configuring your hardware, and you need a right hardware, by the way. And for that, we provide an NGS workstation, which we recommend as an ideal setup for the Flex platform for these kind of workflows. Then there is adaptation of the protocol itself from the manual to the automated. And to make this easy, we have pre-validated protocols in our open protocol library. You can directly download them and optimize them to achieve the data quality that you need. So additionally, these protocols, especially the Evercode protocols, have been verified by the Opentrons and the Parse team. So do you have to be automation expert to use this? Ideally, no. And that's the whole point. And that's why we have built-in automated calibration, robot setup, and intuitive parameter selection, all conducted via the touch screen. This way, even non-automation users in your lab can operate the robot with ease. So we have two NGS workstations, the standard NGS workstation as well as the high throughput NGS workstations. And the key difference over here is that you can configure them based on the needs and the level of throughput you are aiming for. So for medium throughput workflows, you can use the 8-channel pipettes. But if you need higher throughput, there's a 96-channel option available. Now, to further scale up, we have the modular Flex stackers, which you can see in this GIF here. And what it does is it gives you the leverage to use tips and plates, and these serve as storage systems, and they are attached to the side of the robot. And the best part is that you can add as many labware as needed to feed tips or plates onto the deck, allowing you to match the throughput demands of your lab. And then going back to the idea that these protocols have already been written and optimized, we have made them easily accessible to you. And we have an open protocol library over here that's available on the Opentrons website. And it's where you can find the Evercode protocols as shown over here. And from here, you can simply download and configure them to fit your specific needs. Next, to have all of the protocols adjustable. We have, especially these parse protocols, we have dynamically adjustable parameters. And what you can do is you can just have whatever parameter you have in your protocol and then adjust it according to the number of samples, for example, over here shown. So it can range anywhere from 1 to 96 samples. So you simply plug in the number of samples that you're looking for your protocol, and you put it on before you start your actual protocol. So for example, over here, we have 24 samples. You put in the number, and then the protocol will get adjusted or configured according to that particular sample number. And the same goes for other parameters as well, where you can have your liquid level detection on or off. That can be adjusted right at the beginning of the protocol. And the same goes for elution volume, for example, or even the labware that you're using, and the list goes on. Next, what I would like to do is take you through the automation workflow itself. And for that, I'll be specifically talking about the Evercode WT-V3 kit on the Opentrons Flex platform. So to start with, the workflow for automation on the Opentrons Flex is divided into 3 protocols. The first protocol is the in situ cell or the nuclei barcoding protocol, which involves several steps, right from setting up the labware and preparing the deck layout for the actual workflow or the whole experiment to start, and then followed by sample counting. And based on that, we'll have the number of cells loaded onto different, or the first round of the barcoding plate. And so all of these are, once set up, we'll have that experiment started once we upload it onto the Opentrons app, and then the protocol starts off. And next would be the barcoding round 2, and followed by barcoding round 3, and the last step being the lysis and the sublibrary generation. And as Cooper very well explained about all of the sample numbers and as well as the sublibrary lysates that are available for the next protocol, we can have a maximum of 8 sublibraries generated from the WT-V3 kit. And if you look over here, we have the manual process itself, which can take anywhere from 5 hours, 15 minutes to 6 hours total time, with approximately 4 hours of hands-on time. Whereas an automated workflow here on the Opentrons Flex approximately takes 6 hours, 30 minutes to 7 hours, with approximately 55 minutes of hands-on time. Next, the protocol 2 is the cDNA capture and amplification process. And this again involves several steps: the cDNA capture, cDNA template switch, cDNA amplification, post-amplification purification, followed by cDNA quantification, which can be done on the qubit. And here again, the manual versus the automated workflow has been highlighted. And so if you see, the automated workflow will have the hands-on time reduced considerably, and we only have approximately 40 minutes of hands-on time for this particular protocol. Next, the third protocol itself is the sequencing library preparation, which again involves several steps: the fragmentation and end prep, fragmentation and end prep size selection, adapter ligation, post-ligation purification, barcoding round 4, followed by size selection again, and then finally the sequencing library quantification. And here again, the manual process versus the automated process has been highlighted. And I would like to just point out that the hands-on time decreases considerably again to up to only 50 minutes. And then, so the whole of this workflow reduces hands-on time by at least 10 hours on the Opentrons Flex. Next, I would like to talk a little bit more about the whole workflow itself and focus on protocol 1. So here, what we are showing is a deck layout and which outlines the labware, modules, and the reagents used for this particular protocol. And here, what I would like to point out are the modules that are being used, the thermocycler module and the 2 temperature modules at C1 and D1 position. And rest of the labware are in the different deck slots as highlighted over here. And also, the reagents that have been used in this particular protocol have been shown on the right in this slide. And you can clearly see that the samples have been given a color code of blue. And all of these options can be nicely set up at the beginning of the protocol itself. And it's inbuilt in the script. So you don't have to do anything separately, but you can clearly understand what are the reagents being used, where would the samples be in a particular labware. So that is that for this particular slide. And also, I would like to point out that whatever runtime parameters have been included in this protocol have been shown here. And they are the number of samples, which with a default of 8, and they can range anywhere from 1 to 48 for this particular kit. And whether you're doing a dry run or a wet run or a whole live run is highlighted over here. And that again, you can set up at the beginning of the protocol itself. Next, I will go on to protocol 2. So which is the cDNA capture and amplification. Here again, the deck layout has been shown on the left, where the labware, modules, and reagents have been outlined. And then here you can see that there's a thermocycler module at A1, B1 position. There is a temperature module at C1, and there is a heater shaker module at D1, and there is a magnetic module on D2. And rest of the labware, the tip boxes, etc., and the waste chute have been highlighted as well. And if you look at the right side of the slide now, we highlight the reagents that have been used for this protocol, and you can clearly see the color codes for the different reagents being used for this particular protocol itself. And below, you can see that whatever runtime parameters have been used for this particular protocol have been highlighted. The number of sublibrary lysates, the default is 1, but it can range from 1 to 8, as we pointed out earlier itself, because a maximum of 8 sublibrary lysates can be generated from this particular workflow that was mentioned in the slide for protocol 1. And then you also have the PCR cycles that can range from 8 to 13, and that is based on the number of cells that you have been able to count at the end of protocol 1, and that was used for the sublibrary generation. So there is a maximum of 12,500 cells that you can use for a particular sublibrary for this particular workflow. And then based on that number, you'll have the PCR cycles that you would be using for this particular protocol. And then again, you'll have the dry run or the wet run or the actual live run that you'll be using, and that you can conveniently put in at the beginning of the protocol before you actually start the main workflow itself. Next, going on to protocol 3, which is the sequencing library preparation. And here again, we want to show the deck layout that we have used for this particular protocol, outlining the labware, modules, and the reagents. And it's the same as in protocol 2, where you have the thermocycler module, the temperature module, and the heater shaker module, as well as the magnetic block that is present at deck slot D2. And then next, we have the reagents that have been used for this particular workflow, and that's beautifully highlighted by the different color codes for the different reagents that have been used. And then we have the runtime parameters for this workflow, and we see that we have the sample sublibrary that can range from 1 to 8, then the PCR cycles ranging from 7 to 13, and that is determined by the concentration of the cDNA that is obtained from protocol at the end of protocol 2. And then based on that, we decide on the PCR cycles that we would be using for this particular workflow, that is protocol 3. And then again, we'll have a dry run or an actual live run, and that will we can conveniently put that at the start of the protocol. Next, this is the workflow for the Evercode self-fixation. And again, we have the deck layout showing the different labware and the reagents, as well as the modules that have been used for this particular workflow. And on the right, we have the reagents that are being used for this particular workflow. And again, the different color codes determine the different reagents that are being used. And this is a workflow, or this is a protocol that's coming up soon on the protocol library. And I would now quickly shift gears to the data that we have been able to generate from the Evercode WTV3 chemistry. And this is whatever data I'm going to be showing is all showing the automated workflow on the Opentrons Flex. So what we observe over here is that the smooth saturation curves indicate uniform transcript and gene detection across different sequencing depths, suggesting high reproducibility, which is a hallmark of automated workflows. And both of these curves show a smooth increase, and suggesting that scaling up the number of cells or replicates does not introduce any noise or inconsistencies for this particular representation. And both of these curves are showing a clear saturation, and this trend suggests that the preparation, the fixation, and the library prep steps were precisely controlled, which is a key benefit of automation. And going on to the next slide, we see an excellent example of how automation of the scRNA-seq can drive detailed and nuanced insights into the cellular clusters and gene expression dynamics, potentially leading to the identified biological markers. And over here, the genes listed on the left-hand side are all of the genes that are shown under cluster 1. And some of the genes that have been shown here are EDAR, SOC-S3, and RNF175, and they may be crucial in defining the specific functions or the states within cluster 1. And on the right-hand side, you can see the cluster visualization, and we see here that the PBMC samples that have been used for this whole workflow have been visualized as UMaps, clearly showing distinct clusters for all of the different populations of the cell types in PBMCs. Next, we would like to highlight the population of immune cell types that have been generated through this particular whole process. And this UMAP showcases beautifully the population of diverse immune cell types derived from the automated single-cell RNA sequencing workflow on the Opentrons platform. The clear clustering of the cell types, such as the naive CD8+ T cells, the classical monocytes, the plasmacytoid dendritic cells, indicates that automation can produce data of great quality. And if you're curious, just like how I am, you'll see that the spatial arrangement of clusters may suggest relationships or transition states between different cell types. For example, clusters of naive CD8 plus cells and CD4 plus cells are positioned close to each other, possibly indicating similarities in their naive states before further differentiation. Next, we have the expression profile of CD886 marker on immune cell types. So the image depicts the Violin plot of the Z-score of CD86 expression across different immune cell types highlighted over here. And this is generated using an automated single-cell RNA sequencing workflow on the Opentrons Flex. And some of the cell types, like the myeloid dendritic cells, show higher CD86 expression, whereas suggesting a more active role in immune signaling and antigen presentation, whereas other cells, such as the naive CD8 plus cells and the naive B cells, display lower and narrower expression levels, suggesting less activity or different roles in immune responses. So the precision and the clarity of data points across multiple cell types demonstrate the efficiency of automation in handling different samples and ensuring that the data is accurate and detailed profiling of gene expression has been generated through this workflow. Now, summarizing all of the slides that I have been able to show you, this study demonstrates the efficacy of automating the Evercode whole transcriptome V3 kit using PBMC samples, and the whole of the automation process has been achieved using the Opentrons Flex platform. And notably, the generated sublibraries exhibit quality comparable to those prepared manually, and these are compatible with downstream sequencing platforms. And automation of the Evercode WTV3 kit using the Flex minimizes automation operator variability, promoting consistency across all sections of the protocol and library preparations. This integrated solution is particularly advantageous for medium or high throughput projects. Thank you so much. And we are now ready for any questions if you have.
Thank you both. That was great. But like you said, it's now time for our question and answer session. So just to remind the audience, if you want to ask a question, just simply head to the same engagement window to the right of the auditorium and click to open the Q&A. So to kick off, we have an attendee here who's asking, what is the current scope of single cell sequencing in early cancer diagnostics?
Yeah, I can take that one. So right now, Parse is currently strictly in the RUO space. We do have quite a few oncology researchers that are looking at things like tumor microenvironment, tumor heterogeneity for some of these precision medicine methods down the line. I do think diagnostics is a really exciting space for single cell to eventually get to, but I think at this point, right now, we're strictly in that RUO space upstream.
Brilliant. Thank you, Cooper. I think we've got a question here for you, Vasudha. Could you please provide the number of cells utilized in the sublibrary for the data that you presented?
So this is from the WTV3 kit, and the maximum number of cells in a sublibrary is 12,500 cells, and that's what is processed throughout the workflow.
Brilliant. And Cooper, are the WT kits one-time use?
Yeah, so they are one-time use, but upstream of that, you have the flexibility to fix your samples at multiple time points for up to 6 months before you go into the barcoding and library prep portion. Obviously, it makes sense too when you're working with Opentrons that you would automate this as a one-time go. And it also eliminates a lot of the batch effect as well, right? Because now instead of running live cells through the system multiple times, you now get to store those samples, and then compare sample to sample because they were run directly on the same run for barcoding, library prep, and sequencing.
Brilliant, thank you. And one attendee asked if you could elucidate the sequencing metrics associated with the data for SUDA.
Yeah, so the sequencing metrics that are comparable to a manual workflow are the median transcripts per cell, the median genes per cell, the mean reads per cell, and the transcriptome map fraction. And what all of these metrics basically mean are that the very first one, the median transcripts per cell, are the median number of transcripts detected through after carrying out this workflow, and that's per cell that we are able to detect. And the next are median genes per cell, and they are basically, as the name suggests, the median number of genes that we are able to detect per cell. And as well as the next metric is the mean reads per cell, which is again the average reads that we are able to get per cell. And also another metrics which is of importance is the valid barcode fraction, and that's the fraction of the starting FASTQ reads that contain a valid barcode, a set of barcodes that can be detected. So these are some of them, and then apart from that, there is also the transcriptome map fraction, which is actually the fraction of reads with valid barcodes that map to an annotated gene in a given gene model. So these are some of the very crucial ones that we should keep in mind while running any single cell RNA sequencing workflow, especially for automation.
Excellent. And what was the sequencer that was employed for this process?
It's the next-gen sequencing 550 platform.
Brilliant, thank you for clarifying. Our next question is, can you describe the quality control measures implemented to ensure the protocol's scalability, reproducibility, and reliability for further execution?
The very first step when using these fixed cells is, especially for protocol 1, is that the cells would need to be kept cold throughout the whole workflow. Since we know that RNA is not stable and it can easily degrade at higher temperature, so we have to ensure that by active or passive cooling positions which are available for cells and reagents, and for that we have the modules that we use. We in fact use 2 temperature modules in the first workflow, which would keep reagents in that particular labware cool, and for the samples itself which are loaded onto the sample plate, they are kept cool till the time it is actually transferred onto the first round of the barcoding plate itself. So essentially for the first protocol or the workflow, the pre-chilled equipment and consumables are of utmost importance. And also the temperature that is ideally needed to be maintained at for the cells and reagents is between 1 and 4 degrees centigrade. And all of these workflows would require, as far as possible, low bind labware to be used, so that the cells don't stick to the particular labware that is being used during the whole of the protocol. And also one main factor while counting, when we do that at the very first step or before we start off the protocol 1, is to have like sub aliquots of the cells, fixed cells itself, so that we can have the counting done way ahead of the actual workflow itself, so that we don't waste time on that. And also we can quickly accomplish the whole workflow for the automation process itself. And any optimizations to pipetting patterns to reduce the overall time will also help improve the final sequencing metrics. And that being said, at no step would we actually want to vortex the cells vigorously, though the mixing steps would need to be quick, and that is something we are able to achieve using different flow rates during the whole automated workflow itself. And also there is one more benefit, is that because of the different flow rates that we are able to achieve for the pipettes that we use during this workflow. We avoid creating excess bubbles at any of the pipetting steps, because there are a lot of them involved actually in the very first protocol, where there are transfers from the round 1 plate to the round 2 plate. There is pooling of the cells happening. So all of this is very conveniently and seamlessly achieved using automation. One thing to keep in mind is that here we are using PBMCs, but for any other larger cell types, the idea would be to have the pipetting steps done pretty quickly, so that the cells don't settle down when we are trying to have it pipetted or dispensed onto the round 1 plate of the first workflow. So that is that in terms of the quality metrics that, or the quality control measures that would be needed for efficient and reproducible sequencing metrics that we are able to achieve at the end of all of these 3 workflows.
Excellent. Thank you, Vasudha. A question for you, Cooper. To your knowledge, is there any difference in the RNA composition in fixed versus non-fixed samples?
Yeah, that's a great question. What I'll say is with our technology, it's impossible to actually compare live cells to fixed cells in the same system, because we rely on fixed cells. So with live cells, they would not make it through the assay, and we wouldn't be able to barcode. That being said, we have compared with flow, and we've compared with alternative technologies to really make sure that we're not introducing any significant bias. So with flow, we see very similar population percentages across the board with the same exact sample. And then with the technical comparisons with alternative technologies, with fixed versus live cells from the same sample, what we find is we see very high overlap when it comes to the genes and transcripts, but even a larger number of genes and transcripts beyond that high overlap. So if you think about a Venn diagram, there's going to be very high overlap in the middle between the 2 technologies, and then you'll see a larger sliver of parsed genes and transcripts that are only detected with our assay.
Brilliant, thank you. And final question for today's session. Can researchers transition from a manual Evercode workflow to automation seamlessly?
I think that's
yes.
Oh, go ahead, Vasudha.
Yes, definitely. So what we do see is that whether it be the mini kit or the WT kit or the mega kit, there are only few steps where we would have the reagent volumes to be different. Cooper, is that right? The reagent volumes would not differ that much. So we could seamlessly transition from a manual workflow to the automated workflow, irrespective of even the kit that we are using. So if it's going to be a high throughput kit that we would need, and that's what our question is in terms of what we want to achieve, then definitely we can. And Cooper, if you want to add something more to it.
Yeah, and I think really it's a fitting last question, because that's why we're here today, right? I think it is seamless, both all of our different scales of kits as well as our different applications like immune profiling. It's a very similar workflow, which lends itself nicely to automation, because you can optimize. There's always going to be some optimization when it comes to automation, but we've done a lot of the groundwork both at Opentrons and at Parse to validate this and make it very easy to get it up and running in the lab if you do want to automate your manual process with Parse.
Excellent. Thank you both. Unfortunately, that is all that we've got time for today, but I just want to extend a huge thank you to Vasudha and Cooper for today's discussion and presentation. And thank you, of course, to everyone joining us online for submitting your questions. We hope you found that this was a worthwhile session. If we didn't quite get to your question today, we'll follow up with answers to these after the webinar. And of course, if you've got any other questions after this event, please feel free to email me at editor@selectscience.net. Your certificate of attendance will be shared with everyone who's attended live today via email a few days after the event. And if you'd like to listen again to today's webinar or invite a colleague to listen, it will be available to watch on demand. If you're viewing the on-demand version, you can request your certificate of attendance by emailing editor@selectscience.net. So goodbye and thank you everyone once again for joining.